Today we'll be performing the total sulfite test on the EnoLyzer, Unitech Scientific's small wine analyzer. As part of this, we'll be reviewing the use of micropipetters and pipetting technique. Pipetting is a critical step in biochemistry assays. The first step is to set up our materials. The EnoLyzer wine analyzer, the test tubes and test tube rack, the EnoLyzer application sheet, the total sulfite kit from your refrigerator, deionized water that we'll be using for zeroing the EnoLyzer, along with our adjustable micropipetters and tips. You might want to refer to your own EnoLyzer application sheet as we go through the process. We'll be testing a Chardonnay sample and a Zinfandel sample today. And the volume of these samples and the standards is critical, so we'll be using the AP200 pipetter with the yellow pipette tips. So we'll be pipetting 25 microliters of the DI water standards and samples into the reaction tubes. So we've set it for 25 microliters. We're inserting the pipette tip below the surface of the deionized water or the distilled water. We aspirate slowly, bringing the yellow plunger up to the top. As we dispense it into the tubes, we dispense slowly to the first stop, wait for a second, and dispense to the second stop. So let's watch as we dispense the second DI water into the reaction tube. It's important that the sample be completely delivered. If you are too fast in your pipetting technique, you may not get 100% of the sample out of the pipette tip. You'll notice also that we left the pipette tip against the glass tube to draw any liquid out of the pipette tip. Now we're dispensing the sample. This is the 50 milligram per liter standard from our reagent kit. Again, dispensing into the bottom of the tube. Add the standard to the reaction tube as well. And now we'll be pipetting the wine samples, first the Chardonnay and the Zinfandel. Again, when we aspirate the sample into the yellow tip surface of the wine, first we depress the yellow plunger to the load position, the first stop. We insert the tip below the surface of the wine, we release it slowly, then we place the tip in the tube, press it to the first stop, and then just after that to the second stop to make sure all the liquid is out. We're pipetting Chardonnay into the reaction set and just now into the blank set and just now into the reaction set on the right. We'll repeat the same for Zinfandel. Again, we're using a fresh tip for each sample to avoid carryover. First we aspirate Zinfandel into the pipette tip. We dispense it completely into the blank tube and repeat that process for the reaction tube. The final result now is that we have two sets of 4 tubes, the blank, standard, Chardonnay, and Zinfandel. The first set on the left will be getting the blanking solution, and the set on the right will be getting the color reagent from the total sulfite kit. The next step requires that we dispense 1 ml of solution, either the blank or the color solution, into the appropriate set of tubes. We're demonstrating the use of our Eclipse reload pipetting system. So carefully, we take out the 96 tubes, or the 96 pipette tips from the rack. It has a plastic dust cover over the top, so we'll be removing that dust cover and putting it on the remaining pipette tips, and we'll store those for a month or so later when we need to replenish our supply. this is a quick way to do this. You can of course do that manually using our bulk pipette tips. So some people for cost purposes would prefer to buy the bulk tips and not the more convenient Eclipse reload system. We're using the P1000. We securely attach the pipette tip to that. We have adjusted the dial, the blue tip, so that it reads 1000 for 1000 microliters. Now we're using the blanking solution from the total sulfite kit. We put the tip below the surface, aspirate slowly the 1 mil. We are careful to keep the pipette tip above the sample in each case. We want to avoid contaminating the reagent supply, and we want to avoid carrying over sulfite from one sample to the next. If you're uncertain of your pipetting technique, use a fresh pipette tip for each tube to avoid contamination. We dispense blanking solution into the set of 4 tubes on the left. We will find the brown bottle with the color solution in it from our kit, and with a fresh pipette tip, dispense the color solution into the next set of 4 tubes. Again, for the total sulfite and the pre-sulfite tests, these are room temperature reactions, and it's not necessary to use the incubator on board the EnoLyzer. If you're doing enzymatic tests like our glucose or our malic acid or others, it's important that the temperature be at least 73 degrees to body temperature, 97 degrees or 37 centigrade. In that case, we would be placing these tubes in the incubator on board the EnoLyzer, especially if you're doing it in the cellar at 55 degrees, the reactions would be going very slowly. Now we're ready to begin the incubation. The EnoLyzer application sheet indicates 5 minutes, so we've set our timer for 5 minutes. We start. And at this point we're going to turn on the EnoLyzer. It takes a few minutes to warm up, and we're going to select the total SO2 test from the test list window. So the switch is on the power supply, that toggle switch. The read chamber is where all of the samples will be read. So that's and you'll be seeing this. The startup menu shows run test, manage test, settings, and utilities. In most cases, we'll select run test as we are doing now. We look on the menu to find our total SO2. It's not on the first page, so we'll go to the second page. We see that it's test number 11, so we'll select that. I'm going to confirm test 11. So as we're waiting for the 5-minute timer, we can go through the process that we're going to be doing. It's going to be asking us to insert, well, it asks first, do we want to print the full header? To save paper, we're saying no today, but you can print more details on your printout. It's asking the number of samples, and the number of samples we're running today, the Chardonnay and the Zin, is 2. Now the lamp is warming up, and in a minute our timer will begin, or will go off, and we'll start the test. So as the clock approaches 0, we're getting ready to read the blank set of tubes. The EnoLyzer at this point says insert blank. So we'll go ahead and insert the DI water from the blank set of reagents. It takes a while to read, so wait for the EnoLyzer to say remove tube before you take the tube out. Now it says insert blank, standard. Remove tube. Now it says insert sample 1, that's the Chardonnay. remove tube, and insert sample 2. The EnoLyzer now says insert blank, and it means the blank for the reaction set of tubes. So we're going to cycle the blank, the standard, the Chardonnay, and the Zinfandel through as prompted by the EnoLyzer. It's a good idea to be writing on your test tubes the blend ID information. So I usually write B for blank, S for standard, and 1, 2, 3, 4, 5, and keep a list of the blend IDs as a separate. It's now showing us the standard curve. You look at the absorbances. If they seem reasonable, we'll accept. We press the accept button, and now it says insert sample 1. That's the Chardonnay. That result is red. It prints out the initial final absorbances and the parts per million. And for the Zinfandel, again, it's printing the initial final absorbance values and the parts per million. So that's the completion of the total SO2 test. The process is very similar for our other reagents. You may want to look at one of our other training videos for an enzymatic test. Thank you.